The purpose of this experiment is to produce ethanol from pre-treated aspen wood chips. This will be done in two parts. First, the pre-treated aspen wood chips will be combined with distilled water and yeast extract. The mixture will then be autoclaved to sterilize the solution. Enzymes and yeast will then be added, and enzymatic hydrolysis and fermentation will take place simultaneously. During this phase of the experiment, the cellulose in the aspen wood chips is broken down into glucose by the enzymes. The glucose is then immediately consumed by the yeast in a process that produces ethanol, water, and carbon dioxide. For the first phase of the combined experiment, you will need the following pieces of equipment: safety goggles, rubber gloves, heat-resistant gloves, a lab notebook, a pencil, a scale, weighing dishes, a 100 milliliter graduated cylinder, a 250 milliliter Erlenmeyer flask, the 100 to 1000 microliter pipette, pipette tips, aluminum foil, a pressure cooker, a hot plate, an airlock, and a rubber stopper. You will also need the following chemicals: distilled water, sodium citrate buffer, yeast extract, pre-treated aspen wood chips, yeast, Accelerase 1500, Accelerase BG, and 70% ethanol.
Make sure to keep an organized lab notebook. Record all dates, procedures, and results. Wear safety glasses, long pants, and closed-toed shoes. Also wear gloves to avoid contact with chemicals. Ethanol is highly flammable and must be kept away from ignition sources. Be cautious with the pressure cooker and the hot plate to avoid burns and electrical shocks. Handle glass beakers with care to prevent breakage.
The procedure for this experiment is similar to that followed in actual biofuel production, in that ethanol is actually being produced from pre-treated aspen wood chips. Since enzymatic hydrolysis and fermentation are taking place simultaneously during this experiment, glucose is being both produced and consumed. This means that glucose measurements need not to be taken. During the first stage of this experiment, the cellulose in the wood chips is broken down and converted into glucose by the enzymes Accelerase 1500 and Accelerase BG. The glucose is then consumed by the yeast, and ethanol, water, and carbon dioxide are produced. After fermentation is complete, the contents of the flask are filtered and distilled, and the ethanol produced during fermentation is separated from the mixture. In real biofuel production, this process would take place at a much larger scale, and mechanisms would be in place to improve efficiency. The basic procedure, however, would remain the same.
Using the 100 milliliter graduated cylinder, measure out 95 milliliters of distilled water and pour it into a 250 milliliter Erlenmeyer flask. Then, using the 100 to 1000 microliter pipette, measure out 5 milliliters of the sodium citrate buffer and add it to the flask. Using the scale, measure out 1 gram of yeast extract and 10 grams of pre-treated aspen wood chips. Remember to zero the scale after placing the weighing dish on it, so as not to include the mass of the container in your measurements. Place the yeast extract and wood chips into the 250 milliliter flask.
A control solution containing 95 milliliters of distilled water, 5 milliliters of sodium citrate buffer, and 1 gram of yeast extract should be prepared in another 255 milliliter Erlenmeyer flask. This is done to isolate and observe the effects of the independent variable, the pre-treated aspen wood chips. Throughout the experiment, you should observe that no carbon dioxide is being produced, and as no glucose is present for the yeast to consume in the control flask.
Cover the tops of both flasks with aluminum foil in preparation for the next step.
Both flasks need to be sterilized before the addition of yeast to ensure that nothing else is consuming the glucose during fermentation, like bacteria contamination from the lab. Sterilization should be accomplished using the pressure cooker and hot plate. Conduct this step in a fume hood if available, otherwise on a lab bench. First, place the pressure cooker on the hot plate. Add 1 liter of water to the bottom of the pressure cooker and then carefully place both flasks in the pressure cooker. Place the lid on the pressure cooker and turn the seal. Then wait for the water to come to a boil. The water has reached a boil once the safety valve the knob on the top of the pressure cooker begins to rock gently. At this time, the temperature should be reduced slightly and the flasks should be left in the pressure cooker for 20 minutes. Watch the safety valve to ensure that it continues to rock gently, adjusting the temperature as necessary.
After 20 minutes have elapsed, shut off the hot plate and wait for the safety lock to release. This generally happens after about 5 minutes, and the pressure cooker cannot be opened before this time. Once the safety lock has released, remove the lid from the pressure cooker and remove both flasks. Wear heat-resistant gloves during this step, as the pressure cooker and flasks will be extremely hot.
Allow both flasks to cool to room temperature on the lab bench. This is extremely important as the yeast will be killed if the temperature is too high and fermentation will be unsuccessful. Once both the flasks have cooled, use the scale to measure out 20 milligrams of yeast and add it to both the flasks. Swirl the flasks until a uniform solution is achieved. For the next step, the dry weight of the biomass must be determined. The pre-treated aspen wood chips are about 30% dry mass, so 10 grams of wet wood chips yields 3 grams of dry wood chips.
Enzymes should be added to the flasks in the following concentrations. One enzyme is called Accelerase 1500. This is actually a solution of more than one enzyme acting together to break down the cellulose. 500 microliters of this enzyme should be added for each gram of dry biomass. This means that for our 3 grams of dry biomass, 1,500 microliters of Accelerase 1500 should be added. This should be done using the 100 to 1,000 microliter pipette. Our other enzyme is called Accelerase BG. 180 microliters of this enzyme should be added for each gram of dry biomass. Since we have 3 grams of dry biomass, 540 microliters of Accelerase BG should be added using the 100 to 1,000 microliter pipette. Accelerase BG takes the reaction to completion by producing glucose from the cellulose breakdown products.
Sterilize the airlock and rubber stopper with a squirt bottle of 70% ethanol. Spray all inside surfaces and allow to dry.
Fill the airlock to the max line with distilled water. Insert the assembly into the 250 milliliter flask containing pre-treated wood chips, enzymes, and yeast. Bubbles of CO2 should be produced throughout fermentation after a startup period. The purpose of the airlock is to allow bubbles to escape and keep other substances from entering, like air. Either a simple rubber stopper or another airlock can be used for the control solution. The airlock has the advantage of allowing one to more effectively observe the lack of carbon dioxide production.
The flasks should be kept at a constant 35 degrees Celsius in the pressure cooker over the course of several days or until the bubbles of carbon dioxide are no longer being produced. The enzymes need a warm environment to convert the cellulose into glucose, but the environment cannot be so hot as to kill the yeast. Do not replace the lid on the pressure cooker during this time. Instead, simply turn the hot plate on low, place the pressure cooker on the hot plate, add 1 liter of water to the bottom of the pressure cooker, and place the flask in the pressure cooker. Use the thermometer to ensure that the environment remains at 35 degrees Celsius, adjusting the hot plate setting as necessary. If there is concern about the hot plate being left unattended during the day or overnight, the flasks may be left on the lab bench or under the fume hood at room temperature. Although enzymatic hydrolysis and fermentation will still occur at this temperature, enzyme hydrolysis will be very slow.
Once enzymatic hydrolysis and fermentation are complete, as indicated by a lack of carbon dioxide production, the solution can be filtered and distilled. This is the second phase of the combined experiment. The ethanol can be recovered from the fermentation broth during distillation and collected in 20 milliliter glass vials. The contents of these vials will then be analyzed to determine ethanol content, and percent yield of ethanol will be calculated. Careful measurements will also be taken during distillation, including temperatures of the vapor at the top and of the liquid in the bottom flask, as well as of the volumes of the ethanol product collected. For the second stage of this experiment, you will need the following pieces of equipment: safety goggles, rubber gloves, heat-resistant gloves, a lab notebook, a pencil, a scale, a Buchner funnel, 110 millimeter filter paper, a Buchner flask, a sink suction device, a hot plate, a distillation kit, a funnel, a 500 milliliter flat bottom flask, the 100 to 1000 microliter pipette tips, 4 20 milliliter vials, 2 digital thermometers, a 20 milliliter graduated cylinder, and 1 milliliter plastic centrifuge tubes. Once fermentation and enzymatic hydrolysis are complete, as indicated by a lack of carbon dioxide production, the biomass must be filtered out of the flask containing the wood chips. This should be accomplished using a Buchner funnel, a Buchner flask, and a sink suction device. To filter out the biomass, first put the 110 millimeter filter paper into the top of the Buchner funnel and dampen around the edges. With the rubber stopper, attach the Buchner funnel on top of the Buchner flask. After attaching the sink suction device and turning on the tap, pour the solution through the filter paper. Once all liquid has filtered through, first remove the hose from the Buchner flask, then turn off the sink suction device. Only the solution remaining in the Buchner flask will be used for distillation. If necessary, the solution may be stored in the refrigerator until distillation can be started. Using the scale, mass 4 separate vials. Label them with their mass and the numbers 1 through 4. It is very important that each vial remains with its own cap. To record your results, create a chart like this.
Using a funnel, pour the filtered solution from the 250 milliliter Erlenmeyer flask into a 500 milliliter flat bottom flask and add 5 boiling chips.
Assemble the distillation kit as shown and remember to attach the segments together with the blue clips. Cold tap water should be allowed to slowly flow through the condensing column at the top of the distillation apparatus. To begin distillation, turn the heating plate to high power. Once the solution begins to boil, reduce the temperature slightly, but keep it high enough to allow vapor to pass all the way to the top of the column, where vapor will condense on the digital thermometer and the thermometer reading will go up to the boiling point of ethanol. Ensure that the solution continues to boil throughout distillation. Temperatures of both the boiling liquid and the condensing vapor should be monitored at regular intervals, such as every minute. This information will help with assessing percent ethanol in the product. For reference, the boiling point of ethanol is 78.4 degrees Celsius, while the boiling point of water is 100 degrees Celsius. At the beginning of distillation, the temperature of the vapor at the column top should be in the range of 70 to 80 degrees Celsius. The temperature of the top digital thermometer should gradually increase, and distillation should be stopped once it reaches 98 to 100 degrees Celsius. At this point, the vapor is almost entirely water because most of the ethanol has already been separated and collected. Vapor ethanol concentration can be estimated using temperature data and the vapor-liquid equilibrium curve. These estimates can be verified with density measurements, which will be done at the end of distillation. Record the temperature of both thermometers in your lab notebook every minute. Vapor will not reach the top of the condensing column for some time, and the hot plate setting may require some adjustment to ensure that the vapor begins condensing in the water-jacketed column and dripping into the vial. The ethanol produced in distillation should be collected in the 4 20 milliliter vials set up at the beginning of the experiment. Each of these vials should be allowed to fill up approximately 1/4 of the way, then removed, capped, set aside, and replaced. Make sure that the same caps remain with the same vials. This ensures that the mass of each vial itself, without the ethanol solution, is the same at the end of the experiment.
Turn off the hot plate and wait for product to stop flowing out of the end of the condensing column and wait further until the bottom flask has reached 50 degrees Celsius or less. Once the apparatus has cooled, carefully disassemble the distillation kit and pour the remaining solution from the flat bottom flask down the drain. Wear heat-resistant gloves at this time, as the flat bottom flask may still be hot.
Remember to turn off the tap before disconnecting the hose from the water-jacketed column. Complete the charts and calculation of ethanol yield. This is done by first weighing the full vials and determining the mass of the liquid by subtracting the empty vial mass from the full vial mass. Then, take 1 milliliter samples from each flask using a 100 to 1000 microliter pipette. Place the samples into plastic centrifuge tubes and weigh them using the scale. Remember to weigh the empty centrifuge tube first and then zero the scale so as not to include the mass of the container in your calculations. Use the masses to determine the density of the solution in each vial by taking the mass of the 1 milliliter solution and dividing by 1 milliliter. Use the density to determine the concentration of ethanol in each vial, using the ethanol density concentration table. Record all of this information in your chart. Once you have the percent ethanol in each vial, multiply this by the mass of solution in each vial to determine the mass of ethanol in each vial. Add each mass together and divide this number by the initial mass of ethanol, 0.5 grams, and multiply by 100. This will give you your mass balance ethanol yield. Note that if the mass balance is less than 100%, some small amounts of ethanol may still be in the flat bottom flask. A lower mass balance may also be due to other reasons. Can you think of some of the other reasons for low ethanol yield?
In Excel, graph the time versus temperature data that you collected with time on the x-axis and the temperature of the vapor at the top on the y-axis. Since boiling point is directly related to ethanol content, the graph will indicate the purity of the ethanol being collected over time. The purest ethanol would be collected at 78.4 degrees Celsius, and the purity would decrease as the temperature increases to around 100 degrees. How long did the distillation take to collect the ethanol from the bottom flask? Another graph might include the ethanol concentration versus volume of product collected from the condensing column. Is ethanol recovered mostly in the first few vials or the later vials? Comment on your observations.